All right, hey, how's everybody doing? All right, cool. So I was going to tell you a story about some of the efforts we're doing to integrate scientists with the maker community. So where does this really come from? So I'm actually, professionally, I'm a scientist. I just started a research lab as an assistant professor of bioengineering at Rice University. But I'm here because the maker community has been so critical in a lot of the investigations we've been doing in the scientific research lab for bioprinting, for understanding the rules behind building up cells into tissues, tissues into organs. And I want to kind of share with you that story and how we're using that sort of example to unify in a more formal way between scientists and the maker community. So the story starts, I guess we're going to be talking about the Advanced Manufacturing Research Institute that we've been setting up, but it starts in the RepRap community, these open source 3D printers that you've seen around here in the 3D printer village. Some of you may have them at home, some of you may have them at your schools, and this is a fantastic community, right? There's people that are giving you the designs freely and legally. You can download all the designs for this machine, and what we were trying to do is we were trying to use 3D printing in our research. So we're in bioengineering, so we're using engineering principles to understand more about biology. And we're using these 3D printers to understand how to print tissues, how to print vascular networks, how to print blood vessel networks inside of living tissues. And the reason that we use the RepRap and the RepRap community is that all these designs were available to us. People on the internet all around the world were very excited to help us to modify these 3D printers. And so we work with a very diverse team. We work with researchers. This is during my postdoc work. We worked with researchers in the lab at University of Pennsylvania. We worked with our collaborators at MIT, but we actually work with a very large community of open source hobbyists that were developing new software tools, new hardware tools to be able to integrate all these different projects together. And what we were trying to do, so most of these 3D printers that you've seen are printing out plastic, and they're printing out plastic parts. Maybe it's ABS material, the same as Lego plastic, or they're doing PLA material made from corn. And what we were trying to do, we actually wanted to 3D print sugar. And the reason we wanted to 3D print sugar is to be able to make blood vessel networks, something that is similar to a blood vessel network, to be able to harness the nutrient and oxygen delivery system that's already in the body, be able to print something like that, that would allow us to grow cells at very high cell densities. What you're seeing now is a RepRap we modified to print sugar in a 3D structure. And every single filament of sugar that you see there is essentially making a blood vessel network. We can pump blood through, we can pump oxygen nutrients through to keep cells alive. But this talk is really not about this project. What it's about is about integrating these groups. So we were able to get this scientific research paper out about this research. We were rapidly casting tissue modified off of these 3D printed sugar networks, and we could talk more about that afterwards if you want. But what was really exciting about this type of research to me, coming from science, understanding the maker community, is science is really about being open. Science, I'm supposed to be able to reproduce an experiment from someone else in another lab anywhere else in the world. And this goes right in line with the maker community and the open source community, is that all of us are really trying to understand more about just nature, how the world works. We're really here to understand new things about knowledge, like learning knowledge and gaining knowledge and figuring out new ways of doing things. So I had just such a great experience integrating these different communities, given my scientific background, but my interest in the maker community and people just teaching me electronics, teaching me how to modify these RepRaps, that we decided to figure out a way we could formalize this interaction. And that idea, really what we wanted to think about is, what I noticed when I was talking to scientists is no matter how much I told them about, hey, this maker community is awesome, everybody's there, ready to help you, you can just tap into that, you can email them, you can talk to them on IRC, people didn't really get it until we did that scientific study, until we had an example that this is what the power of the maker community allowed us to do in science that we couldn't do by working with a commercial company. Commercial companies wouldn't give us the schematics of their machine to be able to design it, rip out the internals, put on a sugar extruder, recruiter head on there instead. And so what we really wanted to do is think about ways to redefine this experience in a more formal way. So we wanted really structure, instructions for how to do this, and key exemplars for ways in which this could work. So this is how we came up with the Advanced Manufacturing Research Institute, or AMRI. And this is envisioned essentially as a summer program initially, where we're really integrating the maker community with scientists and with the DIY bio community. So some of you may not have heard of the DIY bio community. So DIY do-it-yourself biology. So this is same types of people that are in the maker community where they're interested in learning about how things interact. They're interested in just gaining new knowledge, and they're working with living cells instead of with things like 3D printing and plastic. And so the vision for AMRI is to integrate all of these three groups. And the way that we're doing that is we're focusing on the fellow. We're bringing in individuals to work over the summer. This is very similar to, it was inspired in fact by Google Summer of Code, things like the Blender Foundation where they have summer programs where you have a focused project that you come in during the summer into the research lab. And so in my research lab at Rice University, we had 3 fellows. They'll come up at the end to answer questions and talk about it more. But the goal is that we provide support, and the fellow does this intellectual exploration under the framework, the intellectual framework of the scientific method. And what we realized is that a lot of people in the maker community, they have all of the key characteristics of people that can do science, but maybe because we're doing it as hobby, or maybe we're just doing it for fun, we're not structuring it in a way that we can really make a scientific contribution. So that was the opportunity that we saw, is that if we just structure it a little bit more, then these fantastic people all around the world that are already, they have all the key ingredients to be scientists, we're able to teach them a little bit more about structuring their ideas, structuring their investigations, and make a real contribution to science that we can use for, in this case, bioengineering research. So we had 4 different projects this summer. We ran it in August in Rice University in Houston. And what we did is we found out that there's a lot of really advanced technologies that have been commoditized a bit, and they're available to us for, instead of reinventing things, we didn't have to reinvent the wheel. So we were actually repurposing a lot of these technologies. So light projector, inkjet cartridges, syringes, and lasers. And we realized we could just use these tools, repurpose them for things like bioprinting, biocompatible scientific investigations, and then we were able to adapt that technology for bioengineering research. So we were able to get a lot of sponsorship from a lot of the companies that you see here today. Ultimate Machine was one of our major sponsors, so we're really grateful to that. We got a lot of financial support. We got a lot of hardware support. So a lot of these people, a lot of these companies just sent us hardware that we used during the summer for these scientific investigations. So the framework that we came up with is really in 2 phases. The first phase is engineering design challenge. So this is what do we want to do? What does each fellow want to do? We're going to have a very targeted, very necessary project that they can investigate. So we're going to define, design, and develop the hardware they want to build for the first engineering design challenge. Then after they build something, we're going to measure it, right? So we're going to quantify and qualify. Well, this machine only has this resolution, only has 200 micron resolution, and it only goes at this speed, but are we still under budget? How are we doing in terms of the framework? Is it easy to explain to people how this could work? And then the key part at the end here is to document everything and to deploy those instructions out onto the internet so that other people can reproduce it elsewhere. This goes very much in line with the ethos of the maker community and the scientific community, where we want to be able to reproduce things in other places all around the world. And these are not in chronological order, right? We want to be constantly revising and envisioning again each fellow's project as we go through this. And the vision for maybe for the next year is, after we get this engineering design challenge, after we get a lot of equipment built, then having the fellow, we were just very limited on time this year, but next year we might be able to do actual scientific investigations with the fellows in the research lab, in a bioengineering lab, something like that, where we're applying the scientific method to understand more about nature and more about how we can use this for improving human health in this case. So given that this is a fellowship, we don't want to make people think that this is like a work for hire thing. This is not about you come in, you get paid this amount of money, and you have a device at the end, right? There's no deliverables. What we're focused on here is education. We're focused on improving the knowledge base, the intellectual framework of the fellows themselves. And so this interaction between the scientists and the maker community is the deliverable. We're not talking about a physical final project at the end. Even if we don't get anything done, at least the fellow's going to learn a lot more about how science is done, and we're just trying to integrate these two groups. So what these fellows did when they came to Rice University, they were trained in lab safety training, just very basic. We're using a lot of chemistry, a lot of hardware. It's pretty dangerous in there sometimes, given the scope of things that are in the research lab in a bioengineering department. They also got a crash course in tissue engineering. So a lot of these people hadn't taken biology in years, if not, if at all. So we had a 3-day crash course that is already going on at Rice University that they took part in. And so I'll just kind of go through a couple of their projects, and then we'll bring them up at the end. So our first fellow was Andy Ta. He's over there. He was doing a digital light projection photolithography of plastics and hydrogels. Andy, do you want to say hi to everybody? All right. So he came to us from the Digital Fabrication Studio at Maryland Institute of College of Art. And what he was doing is he was looking at photolithography. So in photolithography, what you're doing is you have a photocurable resin. This is a liquid material that you shine light onto. It induces a chemical change in that chemistry, in that liquid, and that goes from a liquid to a solid. So in this way, you're able to build up in a layer by layer fashion into a 3D object. So what we did is we took this idea, we're going to use photolithography for building up structures. We also want to think about this for biology. We started breaking it down to the core components. So here's one example where we're using DLP photolithography. We're using a DLP projector, the same projector you would have maybe in your home to watch movies on. We can use this for science. So what Andy did is he came in and he repurposed this commercial photolithography device, this commercial projector, and he changed the throw distance of the projector. So instead of focusing 10 feet away, it would focus only maybe 5 or 6 inches away. And we're using this to micro-cure very small areas of this gel. And this is a schematic that he came up with to design this machine. So you can see in the bright blue in the bottom left there, this is the projector mounted vertically, and then he has a Z stage to increment up. So he was able to build this using extruded aluminum parts. He built it very quickly in the lab. And then after he built up the frame, he started integrating all the different components. And you can see some of them here to be able to do this vat-based photolithography curing. And so you can see here so it's 3D printing, right? So the projector provides the X, Y axis for each plane of imaging. And then this aluminum plate is going to increment in Z to pull this object out. So it's going to increment in Z and then project another layer. And the reason that we're doing this for bioengineering research is we want to think about how to build living tissues. And we have some gels, so you may have seen some liquid resins that cross-link a very hard material. We have some liquid gels in lab we can make that we can embed cells into during this polymerization process that is compatible with the living cells, and we can use it to build up structures. So you can see some of the results that Andy got this summer using this type of technology. So in the upper left, that's some of the resin to make that honeycomb structure, and then he started working with these liquid gels that have a very high percent of their volume is water, so it's very good for living cells, and we're starting to look at this for polymerization. So you can see some of the structures he was able to build up in this gel. And we talk about that more at the end. So I want to go on to our next fellow. So Steve Kelly. He's only 19 years old, but he's an undergrad at WPI in mathematics, but he didn't know a lot of biology coming in, but he had that crash course and learned more about the working with living cells. So he was modifying inkjet printer, modifying the inkjet heads to be able to extrude living bacteria and pattern them on a surface. So what he was inspired by is there's a DIY bio group in the Bay Area called BioCurious, and they're a community-based biology lab, and they modified I think it was a CD tray, something like that, to be able to extrude, or sorry, they used I can't remember what they used basically. Yeah, it was like a CD tray, thanks. So they modified that with an inkjet head. They used an inkjet nozzle, and they were able to extrude living bacteria. And what we saw here, you can see they printed with fluorescent bacteria the phrase, "I love BioCurious," but they had a lot of overspray. They had a lot of bacteria they sprayed over the area that they were looking at. So we were going back to the fundamentals. How could we improve on this? How could we improve the resolution and the accuracy that they had? So Steve went all the way back to the fundamentals of how thermal inkjet heads work, how you can heat up a nozzle, you can then induce a bubble formation that extrudes or expels a droplet of fluid in. While mammalian cells cannot survive that, bacteria can. So then he used a rep wrap to give us our z-axis. He used an inkjet head, this ink shield, and connected to an Arduino, the same motherboards that everybody's been using over in the 3D printer village to drive these 3D printers. And the first thing that he did is he had this hypothesis that the height that the inkjet head was off the surface was what was creating this overspray. And so what he did is he implemented this OpenCV program where he's extruding these individual dots in this hexagonal grid. Each of these points in the left side, they're about 200 microns, so about the diameter of a human hair. So very small droplets of ink that he's extruding, and he implemented this software program to characterize each of them. What is their dimension? What is their accuracy of positioning? And how does that all work? And then he went on to develop this software program where you could then draw an image on the computer, import it in, create the toolpath on the right-hand side, that G-code that is sent to the printer for 3D printing, and then do that as a function of height off the surface to be able to integrate many different colors. And then he was able to do some work with bacteria, even though he had no experience with bacteria before. We did a lot of investigations into how bacteria could be deposited onto an agar plate, onto things that they typically grow on. And then our third fellow here today, Andreas Bastian, he was formerly at MakerBot leading the R&D lab over there. He graduated last year from Swarthmore College in engineering. And what he was focusing on is modifying a 3D printer. So very similar to that sugar extrusion I was showing before with melt extrusion of biocompatible sugars, he was interested in modifying this technology using these same materials but finding a different way to 3D print it, and that's based on laser sintering. And so laser sintering is not unlike making a creme brulee. So you can put down a layer of powder, and you can come in with a very hot, either here in this case you have a blowtorch, but you can also use a focused laser beam to induce localized heating of that sugar matrix. So previously during his undergraduate career, what Andreas did is, see if this video will play. Oh, fail. Sorry. So previously Andreas was able to make a laser sintering machine, which you can see here. This was during his undergraduate degree. So this is laying down a layer of powder and then polymerizing that out, or melting it together layer by layer. So what we're interested in is modifying that technology where he was previously doing laser sintering of wax, and could we develop ways to modify that to do laser sintering of sugar to make blood vessel networks, to make the architectures that are naturally found in the liver, which you see on the left, and on the right-hand side, this is a laser sintered part made in plastic. So we were really interested in finding out if that was possible. So that machine that Andreas previously used was a bit overly complicated here. So he had a very low budget in his undergraduate degree program, and this is kind of what it looked like. It was hard to use and hard to modify. So what we did is we looked at repurposing this idea of not reinventing everything from scratch. Could we take a commercial laser cutter, modify it, and then be able to do that for laser sintering of sugar glass. So we just bought this commercial laser cutter. So laser cutters are very good high resolution X, Y, but they have very poor Z resolution. So what Andreas did is he developed this custom powder handling hardware, which you should see here. Sorry about this. He basically made a whole Z-axis that could fit inside of the laser cutter. And we just dropped the bed all the way down. He put this in, it was all laser cut. And then when you put these together, you have one layer, these different pistons, one layer is positioning powder for the next one that is then decrementing down. So you can roll out this layer of powder, you can come with the laser and melt it, and then drop it down a little bit, put down another layer of powder. And he started some of these investigations into the ways that we can sinter this actual sugar together to be able to make the sugar glass for tissue engineering investigations. So I think looking forward, what my lab is interested in is looking at taking some of this 3D vascular architecture and, sorry about that. I guess I could just show it like this. So if you guys can see this here, we're interested in taking 3D vascular architectures from things like the liver, for example. We can take a micro CT scan, we take a CAT scan through that, extract the vascular architecture, understand what that architecture is, and then maybe try to 3D print that in something like sugar for tissue engineering investigations. So anyway, I'll just kind of leave it there. This is still a new program. We're just trying to figure out how this could work, how to bring scientists and the maker community together, and this is still an experiment. So we're very interested in your feedback. What do you all think? Is our framework correct? How could we do this next year? How could we expand this program? And I guess I'll just stop there. We can bring up our fellows to answer any questions. Thank you. Anything at all?
It's okay, I like sitting down.
Well, maybe you guys can just say a little bit about what you thought initially before you came to Rice and what it was like.
Let's start over there.
Start over here? Okay, so go for it.
Oh, yeah. Is it part of transhumanism? How do you mean?
So could you provide a definition?
Yeah, I think a lot of the research that we did this summer was focused on things for bioengineering research. So in my research lab in particular, we're very interested in designing living implants for people to replace a lot of damaged organ function. But this type of framework we think could be general enough that many different areas of science, maybe art, maybe different areas could help integrate the maker community, utilize a lot of the intellectual power that's here, that you see today, and use that for a lot of directed, focused investigations. Oh, the question was about transhumanism, and if this is part of that. Yeah.
In your experiments, you're actually printing a 3D structure with the material that you want the structure to end up on. Have you thought about doing like negatives of the structure and using like a reverse optical sketch? Have you thought about that, or have you tried that?
Do you want to talk about?
All right, so the question was about modeling negative space versus positive space for applications in casting. So in a way that's, we're modeling the positives or the negative space in the resulting gel tissue by, in the case of the laser sintering work that we're doing, we're laser sintering out of a dissolvable sugar material. We're laser sintering that branching tree-like architecture of the vascular network. And then we cast a gel material around that, cure the gel, and then dissolve out the sugar. And so what that does is that leaves these very complex tubular structures that are able to perfuse the entire volume of the tissue. So in a way, we are modeling negative space by modeling positive space first. So it's a sacrificial process, much like investment casting.
Yeah, and I guess I would say we tried both ways. So if you think about, you want to make a tube, right? You want to make something that looks like blood vasculature. So we just start thinking about one tube. So you can think about printing something in the shape of the tube, maybe a cylinder of material, that you then fill around that with gel and then dissolve that cylinder. That would leave a hollow cylinder. Or you could think about the gel itself, printing that one layer at a time and printing everything except for the vasculature. So we tried both of those. So Andreas' project was about printing the sugar material in the shape of the vasculature, which we then would dissolve later in the presence of living cells. So that was a very biocompatible process. And Andy's project was about printing the gel itself, printing everything except for the vasculature. So we don't know which is going to be the best way to do that for making living tissues, but we kind of were trying both things.
So in essence, to figure out which one's best, we're just doing both of them, ultimately.
Yeah.
I'm curious when you say like potential future applications for the bacteria.
Yeah. Do you want to take that?
Yeah. So there's a lot of interesting. Okay, so the question was about what are the applications of printing bacteria? So there's some interesting research into when you combine two different types of bacteria about how they communicate, what kind of materials they would produce, like waste products. And what you can eventually do is, so one of the labs we worked with on a floor above in Jordan's lab, they were interested in making biocircuits, right? So they would try to make like, different logic structures using bacteria. And you can also create like wires. So one thing was that two different types of bacteria will create a wire between them. And if you control the temperature, you can control the thickness of the wire it produces.
If you wanted to access the results of your research and know how you did all the stuff you did, where would you find that?
Yeah, so I guess we're debating right now the proper format for the documentation. So right now, I guess we have a mix of GitHub repositories and Tumblr websites and
Blogger websites.
and regular blogs, and we're trying to formalize that documentation structure. So hopefully in the next couple of weeks, we'll have that all done. But most of the stuff is already online. You can find it linked through the AMRinstitute.org website. And I guess if you have any comments about what is the best documentation structure, so we're not sure if it's like a PDF or HTML or whether it should be an open repo that people will modify later, or if it should be just one downloadable package. We're still kind of debating that ourselves. But most of it is linked off of that website right now. Yeah.
With the 3D printed cardiovascular structures, have you actually ran blood on this to see veins?
Yeah, so some of our early investigations in 3D printing the sugar showed that we could cast gels of any density around there. So you can think about, it's kind of like pouring Jell-O. You start with a liquid material and it becomes a solid. So you can pour this gel material that's biocompatible around the sugar at a very high gel density. And that was it. We showed that it was able to sustain arterial pressures of human blood pumping through it.
And you have a time frame to test it on a living subject? Is that kind of
Yeah, to get to human clinical trials, it'll be many decades. But right now, we're just right now the big challenge in our field is just trying to keep cells alive at very high cell density inside of a big gel. So we can grow any bioengineer can grow literally billions of cells, but they're all on plastic dishes. And when you try to put them together in 3D, they'll all die because they don't have a blood vessel network. So we're just at the stage now we think we can keep them alive longer in a big 3D tissue. So we're not sure about how long they'll survive and whether they have the right function. But we're starting to ask those questions. That's right, yeah. Right now we're just exploring what is the best vessel architecture to make. So I guess Andreas's technology is a lot more scalable to more complex 3D structures than the melt extrusion that we started with. But yeah, we're kind of going on both there. Do you guys want to say anything about...
I think we're out.
Oh, we're out of time? Okay, all right. Thanks a lot.